Hey everyone, how are y'all doing? Good. Um, Hope that y'all had a relaxing break. I know that I posted the videos and today, absolutely, enzymes. So we're switching gears a little bit. We had been talking about oxygen binding proteins, basically dump trucks. Pick it up, move it, drop it off. Pick it up, move it, drop it off. Enzymes will pick something up and then they'll chop it in half and then they'll deal with it. So we're going to talk about enzymes in a couple of different angles. We're going to talk about classification of enzymes. We're going to talk about how enzymes catalyze mechanisms or how they catalyze reactions. So this will give you a little bit of a flashback to organic 2 and maybe organic 1, the tail end where you're pushing electrons around. So just to kind of prime yourself of that or for that, I think that it's helpful to think about electron-rich and electron-deficient species. So something like a hydroxyl group, would you classify that as something that's electron-rich? So it has like a couple of lone pairs of electrons, maybe? Yeah, reasonable. And then something like a positively charged cation, electron-rich or electron-deficient? It's a little bit deficient. It wants those electrons. So thinking about electrophiles versus nucleophiles, if something is electron rich, would you classify it as a nucleophile or an electrophile? In terms of its reactivity, if it's got lone pairs of electrons, it's probably going to be able to attract a nucleus. So it's a nucleophile. Deal? Okay. So that's going to be helpful when we talk about catalyzing reactions. Now, to put this into perspective, typically when you talk about an enzyme, what is an enzyme made up of? Or what's the composition of an enzyme? So, and to frame it a slightly different way, in organic 2, you talk a lot about catalysts, right? Does any catalyst come to mind right off the top of your head thinking of organic 2? Or organic 1 for that matter? Iron as a catalyst. Okay, any organic catalyst? For some reason, for me, whenever I hear organic chemistry and catalyst, I always think Grignard reagent. So what does a catalyst do? It speeds up a reaction, right? It's going to make a reaction go faster, and that's pretty much all that it is. Now, enzymes are examples of catalysts, but they are biological catalysts. Now, biological catalysts are either made up of proteins or amino acids, or what's another example of a biological catalyst? Made up not of amino acids. Like ribozymes? Has anyone heard that term before? So ribozyme is a molecule that's not made up of amino acids, but it does catalyze reactions. So we're largely going to be focusing on enzymes that are made up of amino acids and catalyze reactions with that. So when you think about a molecule that is made up of amino acids and thinking about electrons around, do you think that amino acid R groups are important to know? Absolutely. So something like serine and threonine, those are 2 amino acids that are classified as polar, uncharged, but they also have electrons. So they can use those electrons to move things around. Okay. Now, with that said, what previously mentioned about amino acids and interesting versus not interesting amino acids, which amino acids did I classify as kind of the not interesting amino acid R groups? Your non-polar residues. Leucine, boring. Valine, boring. Isoleucine, even more boring the other ones that I mentioned. So when it comes to like how around, I want you to think about your polar residues, your polar ionizable residues, your polar uncharged, your polar charged, and with your polar charged, positively or negatively charged. Deal? Okay. Now, with that in mind, that's kind of the scope of where we're going. What does an enzyme do? Enzymes catalyze thermodynamically favorable reactions, causing them to proceed at extraordinarily rapid rates. Now, the magnitude of this is something that I do want you to be thoughtful of. Enzymes provide cells with the ability to exert kinetic control over thermodynamic. Essentially, what that means is that an enzyme is a cell's way of basically turning a process on or turning it off, activating or deactivating something. It's the gas and the brake health, or enzymes are the gas and brake health. Living systems use enzymes to accelerate some vitally important biochemical reactions. Enzymes are the agents of metabolic function. And there we go. Now, agents of metabolic function, those are the things that a molecule import a molecule and break it down. So this can of sugar-free Red Bull has 20 calories. So enzymes within my cells are going to take the molecules that are found in this, carbonated water, citric acid, and they're going to break those down into something where my body is going to be able to get 20 calories from that. Okay. Now, if you're interested in that, which you're taking a biochemistry class, so to some degree you absolutely are interested in that, that's what your second semester biochemistry class, your metabolism class, is really going to take a deep dive into. So what enzymes are taking in fatty acids? What enzymes are taking in nucleotides and sugars and breaking them down and getting something like ATP from them? Now, enzymes have unique names. First of all, there's common names. I have no expectation that you remember all of the common names for every single enzyme ever. That would be absurd. There will be a handful that I think are going to be useful for you to remember, not only for this class, but for your second semester biochemistry class and any sort of class related to metabolism. For instance, an enzyme known as hexokinase. That's a very good enzyme to remember the name of, because that is the first step in the process that our cells use to break down glucose. What hexokinase does is it takes glucose and makes glucose 6-phosphate. Okay, so glucose to glucose 6-phosphate, what's happened there? What's the difference between glucose and glucose 6-phosphate? Phosphate. So hexokinase is an enzyme that transfers phosphate onto glucose. And that sets in motion the wheels of change, breaking down that glucose and getting ATP from it, so that we can actually do things with that effectively. Now, examples of common names: urease and alcohol dehydrogenase are two. Urease catalyzes the hydrolysis of urea. Alcohol dehydrogenase catalyzes the oxidation of alcohols and their corresponding aldehydes. These are two enzymes, specifically alcohol dehydrogenase is maybe something that you've heard kind of in pop culture references. Maybe you've heard it referred to as ADH. People who have lower ADH levels, typically if they have a beer or 2 beers or 6, what's going to happen to them is their face is going to get kind of bright red. And they're going to have that tell very quickly. They'll also probably have the tells of slurring their speech and stumbling and everything like that. But from an enzymatic standpoint, alcohol dehydrogenase is going to take ethanol and catalyze it into an aldehyde. Urease and alcohol dehydrogenase are two examples of common names. I do not expect you to know those common names. A systematic name, nomenclature system in which enzymes are named according to 6 major classifications of enzymatic reactions, including subclasses and sub-subclasses. Now, if you're that it's actually not 6 categories, but it's actually 7 categories. Now, the 7th category I'm not going to go into. I'm just going to leave it at these 6 categories. Now, what I want you to think about with respect to this is, let's go ahead and over-under. Who's going to take the over that in all organismal existence, there are more than 5 different enzymes? Who's going to take the over? We got some confused looks, and I'm not sure if that's a confused look of, that's such an outlandish question. Refuse to answer it. But I'm going to go with the over. There's more than 500 different enzymes in every single organism in existence. And you're absolutely right. There are thousands of different enzymes. And then if you consider how an enzyme can be post-translationally spliced or modified, oh man, that makes even more different enzymes. Now, with that said, let's go ahead and say that, let's just give a random number of 12 enzymes known by human beings. Those 12,000 different enzymes are categorized into these 6 different groups. Now, like I said, I'm going to stick with 6 as my number. So I'm not going to do the, hey, well, I say 6, but there's actually 7, into these 6 different categories. So every single enzyme, all of those 12,000 different enzymes, finds their way into one of these 6 different categories. Does that make sense? Now, the classification or EC numbering scheme. Each enzyme also has a 4-part classification number. The numbers represent the class, subclass, sub-subclass, and individual serial numbers. Is anyone familiar with an IP address? Okay. Your IP address has 4 different categories or 4 different, I'm going to use terminology that I don't know well. It's subnet, subnet mask. At any rate, your IP address looks something like this: x.x.x.x. Is everyone good with that? This numbering scheme is similar to that. Now, the reason that I bring up an IP address for your computer or something is if you look at the IP address for your phone versus the IP address for your laptop, if they're both devices that are online, are those IP addresses going to be different? Yes. Is this first number going to be the same? Probably. The second number is also probably going to be the same, and the third number is also probably going to be the same. So you will have these 3 numbers that are probably going to be the same. And the reason for that is because, well, you are getting onto the World Wide Web. So every single device that's online has a different identifier. So what makes your phone unique from your laptop is that last digit or that last set of numbers. So if I told you a specific IP address, you can use information to classify it down and say, okay, well, does it have a number in the first? Or does it have, what's the number right here? Well, that's an indication that it is on the Earth. The next would be maybe a little bit regional, like, oh, this is in North America. Then this last one would be, okay, we can identify this device as somewhere in, Oklahoma, Texas, or Arkansas. And this last one would be your specific device. Does everyone follow that? Enzymes have the same thing. So this would be, oh, well, the first digit represents an enzyme that is going to break a double bond and use water to do so. Okay? The next number would be something like, oh, well, it's preying on this specific substrate. The next one would be, it's preying on this specific substrate with this variable, and then that is a specific enzyme. Okay, so this 4-digit sequence or this 4-number sequence is to kind of funnel down from 12,000 different enzymes to classify them into 6 different groups. And then within each one of those groups, okay, well, this is the substrate that's acting. This is the cofactor that's acting. Does everyone follow that? Okay. So what I want you to take away from this is this EC numbering scheme, 6 different categories. And we will get into those 6 different categories on Friday, but I want you to know that every single enzyme is going to fall into one of those categories. An example would be something like this, 1.1.1.27. And that number is going to mean something about a specific enzyme. If there's 1.1.1.1.28, 0.26, yes. Are those different enzymes? They are different enzymes. What I want you to know about this is not organismal specific. It's not like the enzymes that are in bacteria in my gut and that are in my red blood cells. If it's hexokinase, it's effectively doing the same thing. So the organism it's coming from kind of doesn't matter when we look at this. Does that make sense? Great. Now, there are cofactors in enzymes. These are small molecules that associate with enzymes to facilitate reactions that amino acids cannot do alone. Have we seen an example of something that amino acids cannot do on their own? Or they cannot do well on? Yeah, binding of O2. How does hemoglobin facilitate binding of O2? It uses iron. Well, how does it bind that iron? It uses these heme rings or this heme molecule. That iron is in the dead center of it. That iron helps coordinate and position that O2. Now, cofactors and coenzymes, there are subtle differences in these terms. Metal ions act as cofactors. Copper, iron, all of these are divalent cations. These are all examples of cofactors. Organic cofactors are generally referred to as coenzymes. Examples of coenzymes are nicotinamide adenine dinucleotide, and then a molecule known as coenzyme A. NAD, also known as nicotinamide adenine dinucleotide. Have we talked about, or just hearing that name, I'll say it again, nicotinamide adenine dinucleotide. Do any of those names sound familiar or ring a bell? What molecules made up of? Big class of molecules that we've talked about. We've talked about sugars. We've talked about, what was the other one? Nucleotides. We talked about amino acids. Do you think that nicotinamide adenine dinucleotide has a nitrogenous base in it? Do you think that it probably has a sugar? Nicotinamide adenine dinucleotide. Yeah. So nucleotide versus nucleoside. So we've got a sugar, we've also got a phosphate. So we've got a phosphoanhydride bond there. Now, the molecule, nicotinamide adenine dinucleotide, and I will say that so much that it's one of those words or phrases that you say enough that it feels like it loses its meaning. But I will say that enough until we get to that point. Nicotinamide adenine dinucleotide, NAD plus, is a molecule that's involved in oxidation reduction reactions. Notice how it has that positive charge there? That's by design. That NAD plus. Now, this is a part that I'm not going to kid you. I have to think about this every time I say it. There's NAD plus and NADH. These two molecules involved in oxidation reduction reactions interchange. So NAD plus becomes NADH. NADH becomes NAD plus. Okay, now here's the part that I always have to say and think through. We have an oxidized form and a reduced form. So something loses electrons, something gains electrons. So the oxidized form of a molecule, is that the one that has extra electrons or has fewer electrons? Less. Because oxidation is loss. So if it's oxidized, it's lost electrons. So if something loses electrons, is it going to have a positive charge or a negative charge? It's going to have a positive charge. So NAD plus, is that our oxidized or our reduced form? That's our oxidized form. So NAD plus and NADH, those will go back to the reduced form. The reduced form becomes the oxidized form. That's just the type of reaction that they're involved in. Oxidation reduction reaction. Losing electrons, gaining electrons. Losing, swinging back and forth. And that's one of the things that the first time that we were exposed to oxidation reduction, when was that? Gen Chem 1. So something gaining and losing electrons, that's not really a radical new idea. I think that it's helpful when you see something like this on an exam or a quiz or something like that to take a pause and say, oh crap, Fe goes to Fe3 plus. Fe3 plus, does that have more electrons or fewer electrons than Fe? Be patient with yourself. I was talking with Dr. Britton, our department chair, about a year ago about this, and he was like, oh yeah, I have to do the same thing. And it's like, that's reassuring. But I'm guessing that he's probably doing it like, or it's just like muscle memory. But I will always look at this and I will think it out. Okay, Fe3 plus versus Fe, NAD plus versus NADH. I will do that for myself just because otherwise I will screw up my terminology and I'll be using the wrong thing. So be patient with yourself, do that sort of thing. So I would anticipate a question on a quiz or an exam that is as explicit as NAD plus. This is the oxidized or reduced form of nicotinamide adenine dinucleotide. Make sense? Okay. There are transient or tightly associated molecules known as co-substrates. Co-substrates are coenzymes that are only transiently associated with an enzyme. So that means that they bind and then are released. It's a very temporary interaction. compare that to a prosthetic group. These are coenzymes that are permanently associated with the enzyme. Now, have we used the term prosthetic groups before? We have, in the context of heme. Permanently associated with that protein. And go ahead and feel comfortable dropping this term enzyme and replacing with protein, because hemoglobin is a non-catalytic reaction. So it's going to bind heme and it's going to hold on to that indefinitely. Now, this is kind of a process that you can look at, or everything's a cofactor. But we use coenzymes to speak specifically about organic molecules. Whereas metal ions, those just get the terminology of cofactors. Okay, now this is a table I do not expect you to have committed to memory or anything like that. But I want to give you some examples. Some molecules that you will encounter when you take metabolism is there are a number of different coenzymes that are things that you'll see in your day-to-day life, maybe. So vitamin B12, for example, I believe that's riboflavin, or commonly known as riboflavin. And many of these other enzymes, or sorry, of these other coenzymes are organic molecules that you take as a part of a multivitamin. Many organisms are unable to synthesize parts of these coenzymes, so they have to get them from their diet. These parts must be present in the organism's diet and are called vitamins. Now, looking at some of these enzymes, we will, when we talk about classifying different enzymes, we will talk about different metal ions and the value of different metal ions. When you take metabolism, you're primarily going to be talking about FAD, NAD, coenzyme A. These three things are vital to a cycle named after a biologist whose first name was Edwin. Does anyone know that cycle? You learned about it in a cellular biology class, but as we talked before the class started, we don't have biology prerequisite, but this is something that's the Krebs cycle, Edwin Krebs. FAD, NAD, and coenzyme A are huge products from the Krebs cycle or the citric acid cycle. Because what those can do is those can be fed into the electron transport chain and generate a huge amount of ATP. There's class friends. Okay, so is everyone good with that? Now, what I want you to think about these as is they are involved in some of the chemistry of specific reactions, but also what they can be used for. In the way that I think about NAD becoming NADH is I think about it almost like a currency. Like I'm taking glucose and I'm converting that into molecules of NADH, because that NADH, I can produce a bunch of ATP. Because what do I want? What do my cells want to do? What do I want to be done with all the glucose and fats that I've ingested? I want them to be converted into something like ATP, because I can use that for mechanical movements and mechanical energy. Does that make sense? Okay. So, regeneration of coenzymes. Coenzymes are chemically changed in the reaction in which they participate. The coenzyme must be regenerated to complete the catalytic cycle. In the reactions catalyzed by lactate dehydrogenase, commonly known as LDH, NAD plus is converted into NADH. NADH must be oxidized back to NAD plus to allow multiple cycles of this reaction to occur. So we have this constant movement, this constant regeneration. Does an individual enzyme do this? No, it's a part of a cycle. If you think about the Krebs cycle, for example, I'm going to raise the, no, I'm not going to raise the board. I'm going to give myself I think I can give myself a white slide. No, I'll draw it. If you think about the Krebs cycle, you have input, and then that's basically, yes, I did just draw a circle. Everyone good with that? Okay. Now, if we look at this junction where our two arrows, I don't expect you to know the details of this, but I feel like there's metaphors that are used at weddings all the time. A circular, I give you this ring as a symbol, an unending symbol. When this arrow ends up where I started, what do you think that means from a perspective of your starting point? Like the molecular starting point. Let's say our molecular starting point, if we're bringing 2 carbons from here, and right here, we generate a 6-carbon molecule, well then what has to be where that X is? A 4-carbon molecule. You're absolutely right. Okay. Does everyone follow me there? Okay, so then in the process of this is our starting point where we have this 6-carbon molecule. We go through this to get back to a 4-carbon molecule. What's happening in this pathway? What are we losing? We're losing 2 carbons. Does that make sense? So think about this is a cyclic pathway where we have an input and we have to get back to regenerate one of those components for that starting point. So anytime you think about an enzyme catalyzed reaction, think about it in the same way. Okay, these are my input. My output is this. Well, if I want this reaction to go on again, what do I have to do? I have to get back to that starting point. Does that make sense? So I have to regenerate those components. Now, like I said, an individual enzyme maybe won't do that. But instead, in the citric acid cycle, we've got about 8 different enzymes that are doing it. Enzyme number 1 right here, that is going to feed into enzyme 2, which is going to feed into 3, which is going to ultimately get us back to there, which gets us to our 6-carbon molecule. Does that make sense? Okay. For transiently bound cofactors, sometimes the regeneration is accompanied by another enzyme. That's kind of what I'm summing up with this right here. When we have lots of processes going on to get us back to our starting point. In the case of prosthetic groups, regeneration occurs in a separate phase of the enzymatic reaction sequence. Now, a great example of this, or great models for this, are in the citric acid cycle. Now, I don't have the expectation that you know the citric acid cycle for this point, but when you go on to take metabolism, I want you to be thinking of it. Okay, well, the citric acid cycle, part of it is to extract some energy, but also to regenerate it so that cycle can go on, and this is one of my favorite lawyer words, in perpetuity. My wife is an attorney, so I claim that as knowledge of my own. But this can go on indefinitely, it can go on for forever. That's more my language, for forever, which I don't think is really appropriate language. So when we talk about enzymes, we want to get into and discuss kinetics versus catalysis. Now, when you think about those 2 terms, kinetics, the time. How fast does that reaction occur? What do bonds break and form? Do changes occur sequentially or simultaneously? If you think, Nick, and you probably remember like SN1 versus an SN2 reaction. And there's either furrowed brow that I'd prefer not to think back to organic, or don't think I ever heard those. I'm guessing that I'd prefer not to think about that because you absolutely heard SN1 versus SN2. That has to do with the sequence. Are these 2 happen simultaneously, or does this happen before this? So when we talk about kinetics, we will talk about kinetic mechanisms where we get into, well, this is an SN2 model or this is an SN1 model. We're not going to go too far into that or very far into that, but that is kind of the context that I want you to be thoughtful of. Catalysis, by contrast, is concerned with structural changes that occur during a reaction. Structural changes. If we talk about a protein, proteins have structures, right? They have conformations. Metabolites, or the substrates, those also have structures. Are we talking about changes of one of those or both of those? We're really talking about changes of both of these. So catalysis is concerned with the structural changes that occur during a reaction. How are the bonds made or broken? And what kind of intermediates are formed? Now, what kind of intermediates are formed? That's where we find things like a carbocation or an oxonium ion, where we have these species that are positively charged that, I mean, how often do you talk about a positively charged carbon? Not very often. Absolutely right. I like it whenever somebody gives me the same facial expression that I think I'm projecting outward of a furrowed brow and a no, that doesn't. So we do encounter those as a part of a catalytic mechanism. So we'll talk about kinetic mechanisms and catalytic mechanisms. Catalytic mechanisms are where we find those funky things that Dr. Hudnall's probably concerned and interested in, but I am not that much of an organic chemist. Now, what kinds of interactions occur between the enzyme and substrate products and intermediates are also things that we're curious about. And this would be kinds of interactions between the enzyme and substrate, okay? So whenever I say this, interactions between the enzyme and substrate, what I'm talking about is what types of bonds. Is it a hydrogen bond? Is it a covalent bond? Is it a metallic or is it an ionic bond? Those are the things that I'm kind of interested in. And to dig or to pull one layer back on that onion, interactions between the enzyme and the substrate, well, one good example would be a covalent bond. So a amino acid that's likely or potentially going to form a covalent bond with a substrate would be something like serine. Serine has those lone pairs of electrons. Those are great candidates to drop the hydrogen and make a bond that effectively looks like this. Here's my CH2, here's my serine by default. And now I'm going to erase it. And now I'm going to have my nitrogen to oxygen. Okay. What type of bond is that right there? It's a covalent bond. Nitrogen, oxygen, fluorine, and hydrogen are helpful for hydrogen bonds, but does anyone see a hydrogen here? No. So that's not a hydrogen bond. That would be a covalent bond. And this would be, this serine is a part of my, here I'm going to draw, that's the 3D structure of a protein. Here is my protein with this serine that projects outward. We form a covalent bond right there. So when we talk about catalytic mechanisms, we want to know about, well, does this type of bond form? If the answer is yes, well, then that would be how we classify that mechanism. Does that make sense? So those are the sorts of things that we're interested in. Then we'll dig into that a little bit as we proceed as well. Does everyone feel comfortable with that sort of bond right there? So take yourself back to Gen Chem 1 and Gen Chem 2 and think about intermolecular forces and types of bonds. Largely what we'll be focusing on is hydrogen bonding, covalent bonding, and ionic interactions. So don't stress too much about, oh, it could be anything. Well, it could be anything of those three probably. So proceed. How do enzymes work? Enzymes differ from chemical catalysts in several different ways. Now, one, you're going to get higher reaction rates. And this is higher reaction rates than in inorganic or an organic catalyst. Now, can anyone think of an example of an inorganic catalyst? I'll give you a hint. It's something that typically gets stolen from cars. Catalytic converters, because they have what in them? I know, that's the trick. That's the next level. If I remember correctly, it's palladium. Palladium is where are we? PD. Palladium. PD. Polonium. Neptunium. It's in the middle. Palladium, 46. Palladium. It's expensive. So people are going to steal it. And that's what's in your catalytic converter. Somebody wrote on this. Oh man, some SI is going to get fired. I don't know who wrote on it. It doesn't look like it. It looks like dry erase or Sharpie. No, there's, Dr. Martin ordered them a couple of years. Actually, I think that they were delivered like the beginning of March 2020, right before it shut down. And it was like, hey, we got the new periodic tables. But we're not going to be in class for a while. So we can put them up. Okay. So anyway, higher reaction rates. So if you were able to have an enzymatically driven catalytic converter, the reaction rate would be higher than that of palladium. In addition to that, enzymes are going to provide much more mild reaction conditions, greater reaction specificity, and the capacity for regulation. I want to talk about each one of these things. You kind of did talk about the higher reaction rates, but then milder reaction conditions. Has anyone ever heard of the Bosch, the Haber-Bosch, or the Haber process? It's the exact same thing, but you've probably heard them referred in different ways. This was a tremendous discovery in the late 1800s. This changed, this was like, gave way to agricultural revolution. Yeah, it took N2 and H2 and produced NH3. So N2, nitrogen in the air. What is between those two nitrogen atoms? A triple bond. What does that mean? That's a ton of energy that is stored in N2. Now, taking N2 and H2, you can't just mix them together and be like, well, shucks, I hope that I get some NH3. German scientists took N2 and H2 and I think it's like 700 degrees, and I would not even begin to know what pressure, but under extreme temperature and extreme pressure, we're able to convert those two reactants into NH3. NH3 is absolutely essential for fertilizer. And so it gave this agricultural revolution. Now, all of a sudden, you could get ammonia to environments that previously didn't have good access to it. And so this like less than fertile ground was now very fertile. Now I bring that up because the Haber-Bosch process, super high heat, super high pressure. That is something that if someone was doing the Haber-Bosch process in their kitchen, say that's kind of strange. But also, if you were to go up to that being done and put your hands near it, it'd be extremely hot, extremely unsafe. Similarly, there are organisms. Has anyone ever heard of the phrase rhizosphere? Well, the rhizosphere is basically the bacteria and fungus that grows at plant roots. And there are these things called root nodules that grow at within the roots of legumes. And what they are able to do is they're able to take atmospheric N2 and H2 to produce NH3. So that's why lots of people will do like, if you just grow corn in a field, well, you're going to have to fertilize. But there are some, and that's called a monoculture. What some folks will do is they will have a mixture of different plant populations. So they'll have corn and legumes growing next to one. Those legumes will enrich that soil by doing the same as that Haber-Bosch process. But let's think about those two. If you go to a field where someone is growing legumes, and let's just think about your life experience. How many times have you touched the dirt, compacted ground and been like, hot, 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 hot? Tell me, I'm hearing no. That has never happened. I agree with that. Do you think that's because that process is not taking place there, or do you think that's because, well, the organisms can do that and not go to these crazy high temperatures? It's the second. So the exact same sequence of reactions is taking place, but without those extreme conditions. So the Haber-Bosch process versus what this is known as nitrogen fixation. So taking N2 and H2 and producing NH3 is something that happens in plant roots. And that is just an example of those milder conditions. Now, next up, reaction specificity. If you think about, and the common example that people always like to use, and I'm no different, is I think that it's Tylenol. If you get Tylenol in the United States a 250 milligram pill or something like that. If you get the exact same thing in Germany, it's not going to be the exact same thing. Does anyone remember why? And I'll give you a hint. It starts, it's a fun word, and that is racemic. A racemic mixture. So if you go and you get, I believe it's Tylenol. Yeah. If you get Tylenol over the counter here, you're going to get a racemic mixture. If you go to Germany just to get your Tylenol, you're not going to get a racemic mixture. It's the RS isomers. In Germany, you're going to get a 125 milligram pill because you're going to have a stereo-specific pill. You're only going to get the R, you're only going to get the S. I can't remember exactly. But that's something that's done industrial. That molecule, that is synthesized. And when you synthesize something, you're going to either make a racemic mixture, or you're going to make that racemic mixture and then select for one stereoisomer. Now, if you were producing this with an enzyme, you wouldn't have to do that selection because that enzyme is only going to produce one of those isomers. And then the last, capacity for regulation. And don't think about, oh, well, that's lowercase or smaller letters. That's less important. No, it's not. That's because this was written on a Macintosh, then put onto a PC, and then back to a Mac, and back to a PC over a couple of generations. The capacity for regulation, that is basically metabolism. That is your second semester biochemistry class. Because if we think about the citric acid cycle, yeah, sure, we've got this pathway where we're going to make molecule X, but also different phases in here are going to regulate, like enzyme number 3 is going to maybe speed up enzyme number 2 in some form or fashion based on what it produces. Okay, now this is what I was wanting you to be familiar with respect to the magnitude of an enzyme speeding up a reaction. We have non-enzymatic reaction rate or enzyme reaction. Okay. So let's just take carbonic anhydrase. We have a non-enzyme catalyzed reaction, 1.3 times 10 to the negative 1. Our units there are per second. So think about this as this enzyme goes 0.13 times per second. So in order for this enzyme to catalyze the reaction, like, let's make this simple. We've got carbonic anhydrase is CO2 plus H2O yields H2CO3. So in the absence of an enzyme, mix these two components, and it's going to take about, would everyone agree, roughly 10 seconds for this reaction to be completed once, because it's 0.13 per second. Is everyone good with that? Okay. Now, if we have the enzyme that catalyzes the conversion of this, that reaction is going to be done 1 times 10 to the 6 per second. So let's think about that. That would be 1 times 10 to the 6. Is that 100,000? So in the amount of time that an uncatalyzed reaction produces one of these, so in that 10 seconds, we can do this 1 million times over. Does everyone follow that? With an enzyme. So does anyone see the benefit of having an enzyme? Absolutely. Now, carbonic anhydrase, we talked about that enzyme before. Yeah. CO2 that's generated from cellular respiration in your cells, that CO2 needs to get out. Carbonic anhydrase is going to take CO2, react it with H2O, make this H2CO3 that is more soluble, that can then go through your bloodstream, make it to your lungs. Carbonic anhydrase again is going to do the reverse reaction, producing CO2. So I can't do that. So how quickly would you die if you were relying on the non-enzyme catalyzed reaction? Absolutely. Because think about this, this reaction with an enzyme is done 1,000,000 times over when the non-enzyme catalyzed reaction is done once. Does everyone follow that? Okay, so we look at a rate enhancement. This is just a comparison of those numbers in a specific calculation. This is roughly 7.7 times 10 to the 6th times faster with the enzyme catalyzed reaction over the non-enzymatic reaction. And as you can see, the magnitude of that gets all the way up to 5.6 times 10 to the 14th. When we have staphylococcal nuclease, that's an enzyme that it's from a bacterium known as a Staphylococcus. And that's just their, what is it? That's a genus, species, Staphylococcus, genus. That nuclease is just one enzyme that's found within those staphylococci. And what they're going to be able to do is they're going to, sure, they're only doing it 95 times per second, but the non-enzyme catalyzed reaction is 1.7 times 10 to the 13th. What that enzyme does is it takes DNA and just chops it up. So we have a rate enhancement of 5.6 times 10 to the 14th. Okay. Good. Enzymatic versus uncatalyzed reactions. I think that everyone's bought in. Enzyme catalyzed reactions are a little bit better. They are faster. We're good. Some reactions are so slow in the absence of an enzyme catalyst that their half-life begins to approach the age of the Earth itself. So without an enzyme, that thing's just going to effectively disappear. Enzymes can accelerate reactions as much as 10 to the 17th over uncatalyzed rates. Urease, for example, 3 times 10 to the 4th versus 3 times 10 to the negative 10th. And these exponents, when you see those numbers, it's the same thing that you saw in Gen Chem 1, negative exponent, smaller than 1, positive exponent, greater than 1. So use those numbers and think back to that. The ratio is 1 times 10 to the 14th. OMP decarboxylase, 40 times per second versus 2.8 times 10 to the negative 16th. How are enzymes capable of such extreme catalytic power? Well, what we're going to be able to do with an enzyme is we're going to be able to make these intermediates that very easily fit together that enable us to speed up these reactions. Okay, does anyone have any questions over what we covered so far today? Okay, well then I will see you guys on Friday. We're going to be talking about enzymes. Friday. We may or may not do a quiz. We'll see. We'll see. I'll let you know before the end of today. Deal? Okay. Well, thank you all very much for coming. I hope you guys have a good day. And that's what I got. And I'm going to post this recording on Canvas. And I'm going to try to record these lectures for more often. Stop. Yeah.